Welcome to the Dairy News and Views podcast, a production of the Iowa State University Extension and Outreach Dairy Team. Our podcast covers current educational, research, and industry tools available for your operation to manage healthy cows and calves while producing the highest quality dairy products. Thanks for joining us today on Dairy News and Views from the ISU Dairy Team. I'm Jen Bentley, Northeast Iowa Dairy Field Specialist, and today I'm here with my Extension Dairy Team colleague, Fred Hall, our Northwest Iowa Dairy Field Specialist, as we discuss today's topic with our guest. So welcome back to our podcast, Fred.
Thank you much, Jen.
Yeah, so today we're joined on the podcast with Dr. Chad Dechow, Associate Professor of Dairy Cattle Genetics at Penn State, as he helps us to understand the early onset of muscle weakness defect found in Holstein calves. So welcome to our podcast, Dr. Dechow. And before we get into the topic of discussion, tell us a little bit about yourself.
Well, first off, thanks for having me. It's my pleasure to come and talk about some of the research that we have done at Penn State. And so I am at Penn State. I'm a dairy cattle geneticist, and I've been here since 2003 on faculty. And I grew up on a small dairy farm in New York State, and so I've always had dairy cows, and dairy genetics has been one of my keen interests. So it's been fun to do a lot of research in dairy cattle genetics, and this is one of those projects that I've been involved with.
For the listeners who may not be familiar with the term onset muscle weakness, give us a little background to kind of help us understand.
Yeah, so the condition itself is due to a mutation in a calcium channel in muscles. And so basically the skeletal muscle does not contract the way it's supposed to. And so then a calf can't stand up. And what's interesting about it is that we see similar conditions in humans in the exact same gene. So in terms of where the mutation first originated, the earliest ancestor we've been able to trace it to was a bull named Southwind Belle of Barley, who was born in 1984. And we know that his grandsire and great-grandsire were not carriers. So we think it occurred either in him or maybe in his dam or granddam was the first animal to have it. And he was a very popular sire and had some sons in AI and so forth. But it was at a pretty low frequency in the population until around 2008 and 2010. He had a descendant named Robust, and who was a popular bull, but Robust was the sire of a bull named Super Sire, who was extremely popular, one of the very first bulls in the genomic era that had really high genetic merit. And Super Sire is actually now the most highly related bull to the Holstein population, just to give you an idea how popular he was. So basically, Robust and Super Sire really amplified that genetic defect to the point where animals started to get two copies of it, and that's why we have seen the emergence of this condition.
And so you mentioned just a little bit about, when calves are born, they aren't able to stand. So what happens to those calves? Are they able to live, or do they show signs for the rest of their life? How does that work?
Yeah, so one of the challenges with this condition that we started to realize fairly early on is that this is different from the sense that there are animals that have two copies of this mutation that appear to be perfectly fine. But then others with two copies of this mutation are never able to stand and eventually are typically euthanized. And a lot of times they may perish because they've developed some secondary conditions like pneumonia and so forth. And then there are some of them that have two copies of the mutation that aren't able to stand for a while and then recover, and others that start out their life fine and then develop the condition when they're a few weeks old and never recover. So that's really somewhat different about this condition compared to most when we think of genetic recessives for example, cholesterol deficiency. If your calf is born with 2 copies of the cholesterol deficiency gene, they will never produce cholesterol and they will die. Now, how old they are when they die varies a little bit, but they will all succumb to it. In this case, we call this partial penetrance. So 100% penetrant means that 100% of the time the disease will manifest. And in this particular case, it's less than 100% penetrant. And so we think that probably a little over half of the calves that have 2 copies of the mutation will eventually have to be euthanized because of the condition. And we're trying hard to figure out why some get 2 copies and are fine, and we have not found the magic bullet that allows some calves to survive carrying the condition. We just really have no idea. We've searched hard for other genetic markers that may interact, but have not found anything that's even slightly promising. So we don't know why some animals survive and some don't, but we know certainly this really increases the risk that they're going to not be able to stand up properly.
Share with us a story about how you became involved searching this condition and the reason behind it.
Yeah, so I a veterinarian, a group of veterinarians from New York reached out to me who had a client with some calves that had this particular condition. And they had done some necropsy on the calves and could not find anything wrong with the calves. They did lots of blood work on the calves and just didn't find anything that would indicate any sort of metabolic problem. And so that led them to think, well, maybe this could be something that's genetic. And based on the pedigree itself, there didn't seem to be anything obvious that was a known condition. They had, for example, they had considered, well, maybe these calves have cholesterol deficiency, which was a condition that we've been aware of for a decade or so, but their pedigrees did not indicate that that would be likely. So they contacted me and asked, do I think this could be a genetic condition? And my answer was maybe, but we need a lot more steps before we can really determine that. And so I encouraged them to just send me some samples from the calves so that I could extract DNA. And I told them what I do is I'll put it in my freezer, and if I hear any more people that are having this trouble, then we'll see if we can get a big enough database to kind of track it down. And so from there, I talked to some other dairy producers that I knew were doing lots of genetic selection and that were tracking what was happening to their calves closely. And sure enough, we found some other farms with the condition. And then from there, we were able to build a large enough database to go and kind of track down the condition.
So as you talk about some of the research and the studies that you've done, kind of tracing this back, I believe on the webinar that you hosted a few weeks ago, you kind of talked about this, the story of this random calf. You want to explain that a little bit more?
Yeah, so when we first looked at all of this, everything traced back to those 2 bulls that I mentioned earlier, Robust and Super Sire. And then she was actually a postdoc named Lydia Hardy was working for me at the time, and Lydia is now at ABS Global. And I was discussing this just with my lab group, and she's like, my family's had a calf that is somewhat like this as well. And so we had, it was actually part of an organic study that we had done here at Penn State. And so we had some DNA available from this calf. But when we looked at the pedigree of this calf, there was no Robust, there was no Super Sire. So we really had no idea. It sounded so much like the condition, but the calf's pedigree just didn't match. And so then we started to trace that calf's pedigree back. And that's what first led us to the bull Southwind Belle of Barley, because that was really the only ancestor that was common between this calf and all the other animals that they were working with. And so that turned out to be a kind of an important piece of information, because now we had an idea of the sire that potentially it originated in and so forth. And that allowed us to narrow down the genomic region just a little bit better.
We've mentioned testing kind of as a generic term. What types of testing are out there?
Yeah, so there's two, in terms of the testing that we did to actually find the mutation, there were two steps involved. And the first step was that all the animals, we just genomically tested them. And when we do a genomic test, we tested for about 100,000 DNA markers that were kind of spread throughout the genome. And the point of that particular testing wasn't to find the mutation. It was just to find where in the genome there appeared to be potential for a mutation. And so when we did that, we were able to determine that there was potentially a region on the 16th chromosome in cattle. From there, we then had to do some deep sequencing of some animals and then align that to the 16th chromosome expected DNA sequence. And so we had narrowed it with genomic testing to a region of about 2 million base pairs, or 2 million DNA base pairs. And so we knew somewhere in those 2 million base pairs, there is a mutation that's causing this. And so then we had to do whole genome sequencing. And that's where finding that bull Southwind became really important, because he was an ancestor that appeared that he might have some valuable information in his DNA sequence that we could use. And so we DNA sequenced Southwind, we DNA sequenced a couple other animals, and including a calf that had succumbed to the disease. And that allowed us to, from that 2 million base pairs, to narrow it down to the one base pair that happened to be the mutation itself. Once we found that one base pair that was the mutation, we were able to develop some commercial tests by just testing for that one mutation. And so that is now available from a few different companies. So Genetic Visions ST offers that test, a company called Phoenix Biotechnology, Neogen, they all offer that test, and there are some other companies that will soon as well. And so the important thing for commercial breeders that are just, they're not necessarily marketing genetics, they're just using good AI bulls to advance their herd for commercial milk production purposes, is that the genetics companies and the bull studs, they are now testing all of their Holsteins for this mutation. And any Holstein that carries it, either in most cases, unless they're exceptional for other traits, they're just going to be removed from their population, and they're not going to sell semen from that particular bull. And if they do sell semen from those particular bulls, that's labeled, that's now available on the pedigree, and producers can determine, do I want to use this particular bull, knowing that there is a risk he could transmit this to his offspring. So that's mainly how the elimination of this mutation is going to happen. It's going to be because the bull studs are just removing sires from the population and not selling semen from bulls that are carriers. There are some other tests available as well. So there is now a haplotype test that is available. A haplotype test is kind of an indirect test that comes through regular genomic testing. And so it's based on, rather than testing the mutation itself, it's based on testing mutations that are closely linked physically to the mutation and that are available when we do our regular genomic testing. So when we do regular genomic testing, we're using about 79,000 DNA markers. And so they will test other DNA markers to determine, do we think this could be a carrier or not? So that has some use for herds that are trying to decide, do I have this particular condition potentially in my herd? Is it something that I need to be aware of? Or does it look like I have a pretty low frequency of it in my herd? But it's because it's not a direct test, it's also somewhat less than 100% accurate. So you have to use it judiciously, but it's a kind of a helpful benchmarking tool.
You'd mentioned that Holstein Association is identifying it. How are they identifying it in pedigrees?
So there will be an indication on the animal's pedigree with an MW in the genetic testing region would mean that this individual is a carrier of the condition.
Is that a single or a double? I mean, 2 copies or?
Yeah, so if they have 2 copies, that is indicated with a number 2
Okay.
on there. Yep.
And is this being tested worldwide too?
Yes, it is. A lot of it's being done. Individuals, genetic companies in other countries are actually sending their DNA to US companies that are actually doing the genetic test. But yes, it's being done worldwide.
So in my part of the world, we have a lot of pro-cross and, crossbred animals. How does that affect when we're looking for this new genetic defect?
So if you're doing crossbreeding, the risk of this manifesting is really, really low. And that's one of the benefits of crossbreeding, because the reality is we know that every organism has genetic defects. Some of these genetic defects we know about and most we don't. So this is just one that we happen to find because enough animals that carried it were mated together and generated defective calves. There are other calves that are probably born and that die shortly after birth because of unknown genetic defects that just haven't been discovered yet. So one of the advantages of crossbreeding is that you really minimize the risk of that happening. You don't completely eliminate it because there could be defects that transcend breed even. But for the most part, most defects are kind of within one particular breed. So when you crossbreed, you eliminate the chance, or it seriously reduce the chance that you'll get 2 copies of a genetic defect, and that because of that, you'll never see the condition occur. So that's one advantage of doing crossbreeding.
In my lifetime, I can remember the first genetic defect I was cognizant of, the mule foot. And since then, it seems like it's sporadically, here's something new. Looking forward, the next 10 years, any idea of what's hanging around out there? Is this going to be more and more prevalent to find these as our genetic base narrows, or not so much?
So there are likely to be some others that crop up for sure. And like I said, all of us every organism, including every person, contains genetic defects. That's just the result of mutation over time. We get genes that are broken. And so the real issue is, when do these mutations become predominant enough in a population where individuals are likely to get 2 copies of these, one from their mother and one from their father? A lot of genetic defects actually affect embryonic development, and there's never a calf born because it died, the conception died as an embryo. With genomic testing, we've actually been able to identify several of those, and now bulls are routinely screened for those and eliminated from the breeding population if they're carriers of those, and that's actually helped reduce the amount of early embryonic loss to some extent. But it takes a while and you have to have enough animals genomically tested that should have it to really detect those. So we know that there are some others out there. We know that some others are going to show up. It's just a matter of time and how many. And I think one of the lessons from this muscle weakness condition was that there was a little bit of this idea that, well, with genomic testing, we can screen, we can find these mutations and get rid of them more quickly, because animals that are if it's an embryonic lethal condition, we'll find it. And we kind of relied on genomics to take care of that for us a little bit. And what this has shown us is that even with genomic selection, there are some conditions that don't show up necessarily the first day of life, or that don't result in a stillborn calf or an embryonic lethal condition, that can still pop up a week or two after the animal's born. And so some of those animals may have DNA sampled and genomically tested. So we don't realize that there's a problem until the problem is actually reported to veterinarians and to the breed associations and genetic research community. So one of the lessons from this is that genomics doesn't solve every problem. It makes it easier to discover some problems. We expect these problems to continue to crop up just because that's the nature of genetic selection. It's the nature of the fact that mutations happen. And so that we need to, when we see these things that just don't seem right, it needs to be reported so that we can start down the path of discovery to see, is this really a genetic condition or not?
Well, Dr. Dechow, we certainly appreciate all the great information you've provided here today. Lots of good information, and I really appreciate you explaining, how this came about. And if our listeners want to listen or look more into this early onset, we do have a webinar recording that we'll include in our show notes here today with our podcast. If you want to further view that. As we wrap up here, any last comments, Fred or Dr. Dechow?
Well, it's just, again, I just want to thank you for having me on to talk about this and to just kind of give people a little bit better idea what we have and what we're dealing with, and hopefully this type of condition. I think because we now have a genetic test for it, we are going to see this continue to decline, and we just have to keep our eyes out for the next one to come down the road.
Dr. Dechow, thank you very much for the time. Very interesting material. We look forward to seeing what else is out there.
All right. Well, I don't know if we're looking forward to see what else is out there. We're just prepared when it comes, I guess.
Yep.
Right?
Better said.
Again, thanks for joining us today, and we look forward to visiting with you on the next Dairy News and Views from ISU. This institution is an equal opportunity provider. For the full non-discrimination statement or combination inquiries, go to www.extension.iastate.edu/diversity/ext.